or some sort of mutation load, because every time you duplicate the DNA, there's a chance you introduce, the DNA mutations or whatever it is. And if that kind of something, not completely equal between the sister chromatid is inherited to the daughter cells in a non-random way, that might cause some sort of fate asymmetry. So this idea has been out there in the field for actually about 50 years. And this idea has been highly controversial. And then, some people said, yes, this is happening. Some people said, no, it's not happening. That's been really, controversial. So, and about, 10 years ago, my, the former student decided to really tackle on this question with the best possible way you could imagine. So Sebastian Rapari, that the former graduate student in the lab, came up with a really clever idea how to test this. So she decided to combine, the already existed, the method called the co-fish, chromosome orientation fish, with the strand-specific probe. So let me just walk through how it works. So over here, there's a sister chromatid, and then green strands are newly synthesized strand. So experimentally, you just pulse the flies with a BRDU, so that sorry about that. Yeah. So the BRDU, so that only newly synthesized strand would be BRDU positive. And because BRDU containing strand is 500 times more sensitive to the certain wavelengths of the UV irradiation, you can nick them and then digest them away. That's going to leave two sister chromatid only with a temperature strand. So this is a plus strand and a minus strand, and to which you can, apply the strand-specific probe. One is red color, one is blue color. So you can differentiate which temperature strand was, which temperature strand was going to which side of the cell division, during the stem cell division. And then Swasey also realized you can design the chromosome-specific probe, like X chromosome specific or Y chromosome specific, second chromosome specific, etc. And then with this way, right now you can tell which sister chromatid, like which template of which chromosome went to, which side of the stem cell division. So that means red strand might go to the stem cell, then opposite strand, the blue goes to the differentiating cells, or you can take, you expect to see the opposite strand of the pattern, right, as well. So now, and this is actually example, as I said, you can identify stem cell and differentiating cell at the single cell resolution. Here's a nice pair. If stem cell gets red, and then differentiating cell will get the opposite strand, the blue strand, right? And of course, you can see the flip pattern. Stem cell gets blue, and then differentiating cell gets red. Now, so if two sister chromatids are exactly equal of each other, as we postulated, it shouldn't matter stem cell gets red side or blue side. So these two segregation patterns should happen 50-50 or at random in theory, right? However, when we take a look at the X chromosome or Y chromosome separately, now we saw huge bias over here. And then that means somehow X and Y chromosome, two sister chromatids are not the same. When I say X and Y, we are not talking about the difference between X and Y. Two X sister chromatids, these seems to be different. And then one of the strands seems to be going just a stem cell. And then also Y chromosome, there's a two sister chromatids of Y chromosome. These two seems to be somewhat different. And then one strand is always specifically going to the stem cell. So this bias was really shocking to us because we actually didn't anticipate. We even worried about some sort of experimental artifact or bias because let's say, the blue probe somehow, is too weak. And then whenever we somehow artificially looking at the one pattern preferentially. But then we did the exact same experiment with the second chromosome or third chromosome probe that was completely random. So that means this cannot be our experimental artifact. So we thought, wow, that's interesting, right? And then, but at this point, this was just a fascinating observation, right? But how this is happening, or even more, why this is happening, we had no idea what it might be. That was 2013. And then after that, the new postdoc who took over this project, George Watase, and I talked, and then we decided the only way we can move forward is to actually identify chromosomal element that is contained on the X chromosome and the Y chromosome that regulate this non-random sister chromatid segregation. And then to make a long story short, and then you can tell, that we are actually squishing this 8 years of the story into 30 minutes. So it's a really long story short, kind of, the squishing. So to make that long story short, when George used the X chromosome, that did this huge block of heterochromatin, right? He saw the X chromosome segregation pattern gets randomized. Now it's a 50-50. And then we didn't know which element, and we knew this deleted element contains ribosomal DNA or 359 base pair satellite DNA. We didn't know which is which. So we take a look at the Y chromosome. And the Y chromosome also contains rDNA. That's just specific to Drosophila and Melanogaster. But if you delete this portion, now again, it's also random. That told us, wait a minute, it's ribosomal DNA. And we didn't know what to make of it. And then, in my first slide or second slide, I said, wow, ribosomal DNA is important, right? We knew that much, but we didn't think it's going to be fascinatingly important, right? It's just, at the beginning, we thought that probably is just boringly important. And then now we know that's fascinatingly important. And that we really didn't know what to make of this, but we had to admit that made sense because two autosomes, I said, doesn't segregate non-randomly, do not have ribosomal DNA. On top of that, the George also used the chromosomal translocation where second chromosome got the rDNA, ribosomal DNA from the Y chromosome. This chromosome now segregated non-randomly, meaning, yes, indeed, it's ribosomal DNA, most likely. Now, we wanted to really know, so that allowed us to go into a little bit more of the molecular mechanism. And then, I'm going to cut that. But then, we also identified which sequence element of the ribosomal DNA is likely to be responsible, and which we narrowed down to so-called intergenic spacer IGS sequence. And using that knowledge, we isolated the protein that binds to this IGS DNA. You just, the synthesize the IGS DNA and then put onto the struct avidin beads, and then, the sprinkle stem cell, the extract to identify, the protein that binds to IGS. And then we did the mass spec, and then we did the secondary screening based on the localization. And we identified one protein. I mean, we got some more, but the previously uncharacterized protein CG2199, which we named Indra, seems to be a really good candidate. So we decided to follow up. So first, we found that Indra indeed binds to ribosomal DNA. And, on the chromosome spread, we can see, Indra goes to exactly where ribosomal DNA is. And we also confirmed this by the qPCR. And Indra pull down specifically concentrate this, mostly, the IGS sequence. And now the question becomes, now Indra required for this non-random sister chromatid segregation, that we have to come back to original reason why we screened this, right? And then, so now if you knock down Indra, now this chromosome segregation pattern, the very biased segregation pattern we typically observe with the control X and Y chromosome knock down, the two independent, the RNA lines, both of them randomized both X and Y chromosomes, the segregation pattern. So at this point, we thought, okay, wait a minute, we have such a nice molecular mechanism, right, of biological phenomena of which importance remain questionable, right? So it's kind of at this point that was, the scientist satisfaction, why is it important? That was unclear. So then, okay, we really wanted to know why, the indel has to exist, and then why, the non-random segregation has to happen. And then to understand this, I have to step back and then go back to the intro. I told you that ribosomal DNA is actually repetitive, and because of that, it's highly unstable. And this instability, copy number loss is known to cause senescence in yeast. But not much was known about this copy number and, the aging or senescence in the multicellular organism. So we take a look at the, what's happening in Drosophila germani, and then we first take a look at the male germani. So, okay, so here, the squishing Kevin lose 5 years, former graduate student, into one slide. So Kevin looked at the ribosomal DNA copy number, which is, the plotted on the y-axis. And then, the x-axis is actually the time. And then we know, based on the previous work, Drosophila need at least 130 copies of ribosomal DNA. From 130 to above, typically up to 450 copies, this is a good copy number, sufficient copy number. So as far as you are in this, within this green range, you are good to go. And if you have less than 90 copy numbers in total in the genome, you are dead, okay? But in between, there is an insufficient copy number, but that's not, but still, which is not as low as your, the death, which is, this 90 to 130 copies. That causes some sort of cuticle phenotype. I'm going to come back a little later, but that is a range of the insufficiency. So if you take a look at the wild type flies, typically they are within this green range, the 200, 250 or whatsoever, right? But if you follow these flies and then his germline, we could see that copy number actually goes down during aging of this male, right? And then, as I said, this is happening in the germline. What does that mean, right? If germline lose the copy number, of course, that's going to go into the sperm and then go to the next generation. So that means this son from the older father end up is inheriting reduced copy number, right? And then here you see the problem. If this keeps happening, if this son gets older, he's going to lose the copy number. And then if this keeps happening in a few generations, you completely eliminate the kitchen pest program. But at the same time, we will be going extinct together too, right? So that's a huge problem. Why this is not happening yet? So then what we realized is this son might be starting his life with a reduced copy number. And of course, somatic cells, there's nothing they can do about. However, his germline, again, somehow be able to recover the copy number so that grandson is now okay, right? So he has a nice recovery, the process. And then that is probably what is keeping us and, the flies from extinction. And then when we saw this, we immediately realized, hey, there's an old literature. That was, from 50 years ago. People have been using this interesting flies that is within a range of insufficient copy number. And this guy get the cuticle phenotype. But then, upon induction, if you try to, minimize the copy number of this guy even further, then somehow there's some sort of sensing mechanism kicks in, and then he can actually recover the copy number in the germline so that the next progeny is now in the okay range. So this phenomenon has been called rDNA magnification from 50 years ago, right? which is, but then back then, this was more like a pure pedantic interest, the academic interest. Like starting from weird chromosome that doesn't have enough copy number of the rDNA, somehow it recovers. It's interesting, but what does that mean, right? Now we think it's not just this is about the weird chromosome program. We think this supports this fluctuation of ribosomal DNA copy number to keep us immortal, to keep our germline immortal. And indeed, we found that, the certain genes that's been known to be required for this magnification is also required for this recovery in the normal fluctuation. So that brought us to hypothesize, the magnification is not just a, weird phenomena. It's probably a manifestation of the regular maintenance mechanism. And now, if that's the case, and we think that's the case, the magnification and regular recovery, they have a parallel, and then, the shared molecular mechanism. Now we can go to the mechanism of the magnification. So how do they magnify the ribosomal DNA, right? And then, so this is how they do. So here is a sister chromatid of ribosomal DNA after the DNA replication, right? I mean, there's you have two copies after S phase. And to magnify, this is what they do. First, they undergo an equal sister chromatid exchange, meaning these two sister chromatid misalign on purpose, okay? And then still you can find the homology locally because this is a repetitive DNA. And then you undergo the exchange over here, DNA recombination here. And then what happens if you resolve this, right? This is what happens because you go this way, exchange, and then continue. You can see here is an asymmetry in the copy number. And then we realize this is the asymmetry perhaps we are looking for when we saw this non-random sister chromatid segregation. Right? And then we think, and then, so I'm going to show you the actual evidence. We think stem cell is getting this bigger copy. Okay. So now, first, I want to just go through, the basics to cover the basics. Is indra required for the ribosomal DNA maintenance, right? I mean, that's the only way how things make sense. Indeed, if you mutate the indra, this is what happens. So once you establish your indra mutant stock, right? The first generation still, that they start losing the, the fertility compared to the control. And the control, of course, no matter how many generations you follow, their fertility is okay. I mean, that's how, the fly have not gone extinct yet. So, so is us, right? But, indra mutant keep losing fertility. By F2, they are essentially gone. And then during that time, rDNA copy number also decreased, and then we use this digital droplet PCR, which is fairly accurate in counting the copy number. And this ribosomal DNA copy number goes down upon, the mutation of the indra, depletion of the indra. Okay. On top of that, we also could do the rDNA magnification assay. You start from this insufficient copy number, and then, when you match, the G chromosome, which is actually Y chromosome, also, the forcibly reduce the copy number, the fly realize, okay, we don't have enough. And then, this magnification mechanism happens, kicks in, and the next generation, you observe this, the magnification. And then this is actually the phenotype you score. If you don't have enough copy number, you see this cuticle is not just straight. And then this is, the well-established hallmark of rDNA insufficiency. And compared to the normal, the nice cuticle, the nice stripes, parallel stripes. Okay, so then, So then if you look at the regular flies, but then non-magnifying conditions, that means you are not inducing the magnification. They barely recover. But if you induce the magnification conditions, like more than 20% of the progeny recovers, right? To go back to this normal cuticle. you give the exact same magnification conditions, but also deplete Indra. Now you don't magnify. So that means, indeed, Indra is also required for the rDNA magnification. So, it's required for the ribosomal DNA copy number recovery. So, okay, now I'm showing exact the same things again. This unequal sister chromatin happens, right? Exchange happens. Can we see it? That became the really final focal point for this project. And we really wanted to see it. And then at the beginning, we thought maybe we can see this copy number difference in the dividing germline stem cells, right? And then, again, it stopped progressing. Let me try this. Okay. I think I should actually show you this one first. Yeah. Okay. So at the beginning, we just did this anaphase, the germline stem cell, and this is going to the stem cell side, this is going to the differentiating side, right? And then this is not the condition that's increasing the copy number. But when we did that, we didn't see any copy number difference visible. And then we were very disappointed. Maybe, the copy number difference is not enough to detect cytologically. And then, we were disappointed, but then, there's nothing we could do. But this later on served as such a perfect, the control experiment, because we wanted this to be asymmetric, and then we didn't see it, right? So we, if we could have been biased, we wish this would be more. And then, despite our wish, we didn't see it. So we are sure that this does not happen, right? But one day, the George realized, hey, maybe we are looking at the wrong condition. And then he looked at this magnifying condition when overall copy number is so low that this magnifying, the mechanism is kicking in. All of a sudden, we do see this asymmetry. Always more is going to the germline stem cell. And I'm sorry, animation is really behaving. Now my mouse is behaving a little weird. Okay, so here, this is what it is, right? Normal condition, non-magnifying condition, nothing happens. The germline stem cell differentiating side is completely, the 1-to-1 or, the same ratio. Log 2 is 0, meaning 1-to-1. But if you induce the magnification, now germline stem cell gets more, and then this is statistically significant. Interestingly, if you mutate Indra, also it's a magnifying condition because it's overall low ribosomal DNA amount. Now it goes back to 50-50. So this comparison makes us pretty confident. Yeah, this is what's happening, right? The germline stem cell is getting more. And then let me go back. Yeah. Can we actually see, is this really true? This exchange actually really happening, right? To explain more germline, more ribosomal DNA on the stem cell side and then fewer on the differentiating side. So can we actually see this exchange? That became, the really final, the inquiry. And then, so this is a chromosome, the X chromosome. And then when we were originally checking the chromosome segregation pattern, we are using this probe, the proximal to ribosomal DNA. And the red strand goes to stem cell, blue strand goes to, the other side. There's another satellite DNA we could use, opposite side of ribosomal DNA compared to the original probe, right? And then when we take a look at that, typically this also, this also goes to the stem cell side, which you can oversee here, right? TAGA side, 359, both, the red strand goes to the stem cell side. That's what we see. However, once you give them the magnifying condition, right? 359 stays equally, the biased. All of a sudden, the other side flips or, doesn't segregate, in a biased way. That can be only explained if exchange happen over here. inside the ribosomal DNA because, the sister chromatid flips this way, right? That's what we are observing. And so that's something we're super, super excited to finally discover. Okay, so with that, so I showed you this. And then, starting from this asymmetric cell division, and then following up on the non-random sister chromatid segregation, we think, that we found something really related to the germline immortality, right? So in our system, at least, this non-random segregation is not really about the epigenetic difference or mutation load. We think it's a ribosomal DNA copy number. And then that copy number difference, and then that is bounded by Indra. That explains or that mediates non-random sister chromatid segregation. And that is the key to maintain germline immortality, okay? And then, actually, I didn't explain what it means, the why we named it Indra. So this is Indra is a Hindu god, and we had an entire lab meeting one time, a few years ago. How do we name it? And then, somebody brought up this name, Indra, because this is a god. Normally, God is immortal, right? But because of the curse, this god, Hindu god Indra, became mortal because of the curse. And then, the Indra mutant, the following Drosophila, the tradition, you're going to name the gene based on, the mutant phenotype. And the Indra mutation makes the flies mortal, the germline mortal. That's why we named them Indra. Okay, so in the last, the last 10 minutes or so, I want to just go over another story that's related to the ribosomal DNA. And then, I already told you this unequal sister chromatid exchange is the key, for the copy number recovery. One thing I didn't really say was how this is initiated, right? To induce the recombination, you have to first break DNA over here. How this is happening. And then, so this is a work by, the Jonathan Nelson, another postdoc in the lab. When he started, he was really interested in this transposon called R2. This is a retrotransposon that specifically inserted into ribosomal DNA. And then typically, we all think that, all those retrotransposons are parasites of our genome, right? But then, all those parasite goes to most important part of the genome specifically, how loser it has to be as a host, right? I mean, your most important part of the genome is infested by this, the parasite. How come as a host should you allow that to happen? And then, the Jonathan felt that maybe that's not what it is. Maybe actually R2 is inserting here for a reason, right? And then, so he decided to take a look at the, what the R2 retrotransposon might be doing. And then when he, I don't want to click too many times because it goes so many times. Okay, so yeah, there you go. Okay, yeah. So first, when he knocked down R2, and then, the surprisingly or half expectedly, it lost the RDNA copy number. Okay, and then, again, this is a digital droplet PCR, and in mutant situation over here and here, over the aging compared to the wild type, it's going to lose the ribosomal DNA copy number quite a bit. But that is also, reflected to the I think I skipped. Okay, so I'm really sorry. I don't know why my mouse just keep going like stupid way. Yeah, okay, this is the first. So knockdown of the R2 actually reduce germline stem cell, the number, like dramatically, right? I mean, compared to the wild type during aging, the germline stem cell keep losing, they the reduction in the germline stem cell number is really, really, the profound compared to the wild type. And then, looking into the cause, we found copy number is decreased. And indeed, this is because of the ribosomal DNA copy number. Because if you, this is pretty crazy, I'm sorry. If you supplement ribosomal DNA copy number by adding a mini chromosome, then, the R2 knockdown mediated the stem cell number loss gets completely rescued, okay? So that means R2 seems to be required for the stem cell maintenance through the ribosomal DNA copy number maintenance. Okay, so how R2 does it? And then probably by now you might have guessed, somebody has to make a fast DNA break to induce the copy number recovery. And then because retrotransposons, its job as a parasite is to hop into the genome, right? When they are hopping into, actually you make the DNA break. So we thought that might be that. And then indeed, if you overexpress R2, this is a proof of, the principle. You can see that, the R2 breaks ribosomal DNA. And then, it's might not be showing, but this green is ribosomal DNA, and the chromosome breaks at the ribosomal DNA. So this Y chromosome is broken apart where the ribosomal DNA is. Okay, so then, now the question is, when R2 is expressed, because retrotransposon, even if it's somewhat functional or beneficial, you are not going to express all the time, because if you do so, totally parasite is going to win, right? So then we actually wanted to take a look at when it's expressed. So then when we looked at when it's expressed, it's actually specifically when ribosomal DNA copy number goes low. So in a normal condition with sufficient rDNA copy number, we don't see many of the germline stem cells express R2 at all. But once you induce the so-called magnification I told you, which is a low rDNA copy number, that means they are in need of expanding ribosomal DNA copy number. Now many germline stem cells start expressing R2. Okay. Now, is this the cause of the double strand break you're making? And indeed, it seems to be the case. So in the normal condition, germline stem cell, the encircled over here, they barely have any double strand break, that's marked by gamma HCAB, and then, he's scoring like only 4%. But if you do the magnifying condition, that means, the low rDNA copy number. Now this goes up to the over 10% of the germline stem cell at any moment have 10%, more than 10% of the germline stem cells have, the double strand break in it. And if you knock down R2 on top of this condition, you end up with suppressing this double strand break. So that means when cells are trying to magnify ribosomal DNA copy number, R2 gets expressed. And that is how you are, making double strand break. That's R2 dependent. Now, of course, we wanted to know then, do we need R2 to keep going, right? Because if it's required for ribosomal DNA copy number maintenance, and then of course it got to be required for the germline immortality. So this is the assay John did, and again he made the stock that constitutively express R2 RNAi construct. In case of control, of course, all those lineage keep going, they survive, right? But then if you, make, the R2 RNAi stock, as they go on to the next generation, the stock keep dropping. And then, the individual stocks actually, they start going, the extinction. So that's what we see. And then when this is happening, indeed, we do see the rDNA copy number loss. It's not that much, but this is, the combined with actually extinction. That means if, copy number goes actually below certain things, before we can detect it, right? I mean, they are dead. So we cannot even do the, DNA extraction from non-existent flies that's already dead. That's what we think. So to summarize, right, this is actually the fluctuation of the rDNA copy number. The young flies or certain flies have a long enough copy number. As you age, you're going to decrease the copy number. So that is the one problem of the aging. But then, you cannot keep this, the happening. So then that means at some point when copy number decreases, right, you have to have, the R2 heterotransposons to come in. Sorry about that. Yeah, come in. And then, that actually helps the recovery of the copy number. Sorry, I didn't talk about R1, and then I should have updated. We actually don't think another heterotransposon R1 does this job. So I shouldn't say that, that it's only R2. So then, this is really unexpected function or benefit of the heterotransposon, not just junk. So that's what we see, we think right now. And yes, indeed, as a summary, I hope I conveyed that, unexpected mechanism of the germline immortality is a rDNA, that repeats that, inevitably always keep decreasing during aging, right? And then when that happens, you have to recover, otherwise you're going to go extinct. So then, what they do in a germline is they induce double-strand break so that they can undergo unequal sister chromatin exchange to increase the copy number, right? And then copy number gain. So, and then for, this formation of the double-strand break, the R2 heterotransposons have a very critical function. So unexpected functionality of what people believe to be a parasite. And then this asymmetric segregation of the sister chromatin, and that's where, the indra is functioning. And then this gained copy number, the increased copy number specifically goes to the stem cell, and that's mediated by indra. And then, because, the stem cell inherit the recovered copy number, the one big benefit is that, such a stem cell can generate many, many more sperms with a recovered copy number. That's what we think is happening. And then, so, the first part of indra was primarily done by, the postdoc George Watase over here. And the second part of, the R2 and, copy number recovery is done by Jonathan Nelson. He's on the job market this year, and George is going to be on next year. And then, other than that, I'd like to thank all those funders and the support and the Fly community, etc. Thank you very much, and happy to take any questions you may have. And sorry about this mouse mishap. This always happens with me somehow. Sorry about that. Okay, great. Thank you so much.
All right. Thank you, Yukiko. That was great. So if anybody has any questions, you can just feel free to unmute yourself and ask away.
I have a question. I'm sort of curious about, seemed like there were, I have two questions actually. So the first is, there are the few cells where you get the larger one going to the differentiated cell, because there's always like a small percentage. Yeah. Did you track those? Is there any benefit then, or do the stem cells lose their stemness?
Yeah, that's you have a pretty good eye. And then let me try to go back, can I? Always. Oh, sorry, maybe I should escape. And then, yes, you are really good with observation over here. Yes. So sometimes, this goes, how to say, the actual opposite happens in a few cases, right? We don't know why this is happening, and this is intentional, or we actually don't know. That's a really good point. But then, at least one important observation for us is that we are fascinated that in this condition, right, when nothing is happening, and then, the cells have enough copy number, this 1-to-1 is pretty in a narrow range of 1-to-1 over here, right? So that means there's no unequal sister chromatid exchange really happening. And then this, that they are really within a tight range. But once, the low, that we induce this magnification by the raw rDNA copy number, now this range gets really wide. That means, the imbalance where differentiating cell gets more, this normally doesn't happen. This normally doesn't happen in a wild or normal situation. So that means at least they know to exchange, right? Undergo unequal exchange. And after that, they seems to make a mistake and they flip it, or I don't know, there might be actually the really good reason for it. But we have not been able to track it down, so we don't know. But that's a really good question. Yeah.
And then my second question was sort of, can you co-opt this in somatic cells to maybe, prevent senescence, or has anybody tried that?
That's a really good point. We don't know about Drosophila somatic stem cells because Drosophila lives only like 2 months, right? But I think we believe we can draw the parallel between this and then also telomere. And the telomere, if you look at the mammals, telomerase is most active in the male germline because you have to really increase the telomere to bump up, right? And for the next generation. But at the same time, the mouse are long living enough so that their somatic stem cells, such as, hematopoietic stem cell, also have to upregulate the telomerase to some extent, not as much as in germline. So that means I think any stem cell that's living long enough, I feel they should have the ribosomal DNA copy number maintenance mechanism. So I think that's something I want to really go next. But at the same time, again, same things as the telomere. If you upregulate the telomerase, that could actually lead to the tumorigenesis, right? So there might be the balance of how much you want to do, to activate this, the magnification process in a stem cell, because that could be the, another cause of the tumorigenesis. Yeah.
Thank you.
Thank you.
Yukiko, would you predict R2 expression in BOBD mutations?
Yes, actually R2 gets highly upregulated in a BOBD condition. Yeah.
Okay.
Yeah.
So as I mentioned to you earlier today, we see R2 expression quite heavily with the depletion of a ribosome biogenesis factor.
Yeah.
And so, we see a loss of ribosomes and then the induction of R2. So whatever the mechanism is to count rDNA copy number, maybe it's the production of ribosomes that
I think
R2 expression.
Yeah. I think that's really, the good point. And then indeed, ribosomal DNA copy number might be monitored by actual production of the ribosome.
Yeah.
That's quite likely. Yeah.
Right. But how that works, we have no idea, right?
Yeah, exactly. Yeah.
So, a lot of work to do.
Yeah, and I think that's, the one interesting thing is, what was that? There was some other ribosomal component that cause, the low abundance of the ribosome. That does not necessarily induce the magnification. So that means, the NOP140 might be in an actual special spot.
Yeah.
You know what I mean? Not just the ribosome number that's being monitored. Maybe NOP140 is actually the part of this pathway that really monitors, quality or quantity of the ribosome being
right.
made.
Right.
Yeah.
There are predictions that NOP140 actually interacts with PAL1. So it's in there, right by the rDNA.
That's right.
So minute mutations that particularly knock out ribosomal proteins, would they affect R2 expression?
Sorry, which?
The minute.
Yeah. Oh, we haven't looked at it. That's really interesting. Yeah.
Okay. Thank you.
Yeah, thank you so much. That's really insightful. I think we should take a look at those things. Yeah. Okay.
Yukiko, I thought it was really, really interesting. I have two sort of just like conceptual questions that are related to what Alyssa was asking you with that last point. And I know you said you were sort of wanting, this is something you're thinking about where you want to go next in terms of thinking of the application. So my first question for you is, because I was thinking of this same distinction between like the soma and the germline. And of course here, you're showing the male germline stem cells. I was just thinking if you all have thought or done anything specifically with the female aspect of this, or sort of the female germline, or is there any expectation based on what you know that it would be different?
So I would have thought females should be doing the same thing, right? But historically, this magnification, when people are just using this mutant chromosome, right? Magnification doesn't happen in a female. I don't know why. Okay, and then people even mapped, if you make XXY female, that female undergo magnification. It's crazy. I don't know how to make of it, how to think of it. But that, so that means something on the Y chromosome seems to, be regulating this process of magnification. Why female doesn't do it, I don't know. Yeah.
But those repeats are also on the X.
Yeah, exactly. Yeah. X chromosome also have a repeats, right? And then, the repeats actually magnifying in the magnifying male is the one on the X chromosome, but the female doesn't do it. I don't know. One, the far-fetched maybe speculation is female has 2 X chromosomes, right? I don't know, but then what's wrong about the recombining between 2 X chromosomes, right? Because recombining between X and Y would be even worse, right? But I don't know. But yeah, I think why female potentially might do it differently, that is actually a really unsolved question. That's really important, I think.
Okay, so this actually ties into one of my other question unexpectedly, because then I'm thinking also, because you're mentioning about sort of germline mortality, but I was thinking of it more from a perspective of like reproductive aging, if that makes sense, right? Because they're sort of two distinct things.
Yeah.
And I'm just curious and thinking like, so you show that these mutants that have a loss of function in this process, they sort of, the lineage will die out, right? But if you had something that was a gain of function in this process, would it necessarily maintain like fertility or reproduction like later, or would that not really be a benefit? It would just be that the line's maintained, like the lineage is maintained.
Yeah.
I hope I make sense, but I'm just...
No. Yes. I think, the similar version of the question is, if germline have ability to recover in the next generation, why do you age in the first place, right? So I don't know. So one thing I could think of is when this non-random sister chromatid segregation is happening, that depends on actually functional centrosome, right? So then we know that during aging, some aspect of the centrosome changes, and then actually that is, in the early germline stem cell, early on in the young flies, always this really old maternal, the mother centrosome, all this centrosome with a little bit of higher microtubule, the organization ability is always sitting on the stem cell side. And we know that is required to grab the correct the DNA strand. So then we also know during the aging that all the centrosome gets lost. So now one thing we are wondering is, this recovery mechanism is affordable only very narrow time window of the early germline. And if that's the case, when you are aging, you cannot activate this recovery program because you are incapable already. But once you do the fresh start in the next generation, there's some time window, first a week or two, they could recover. But again, with that said, that this is a pure speculation that I can't really say anything for sure.
Yeah, I guess then sort of another point branching off of that I was just thinking of is, a few of these systems, I know they, people will say like a big determining factor of this are these long-lived proteins. And I was just curious if any of these proteins involved in this like fall into that sort of category.
Yeah, I don't know. I think we have been interested in, long-living proteins or, also the opposite way, the denatured proteins you actually want to get rid of, right? How we haven't been able to detect anything in our system, but then it's also possible that, we didn't look hard enough, right? Because those kind of things, unless you know what's going on, you can't even, generate the reporter or, the way to look at it. So, the typical things, if you're just looking at for example, the Huntington proteins, that's going to, the aggregate, etc., you overexpress it, we don't necessarily see any asymmetric inheritance in our stem cell division. But, it doesn't mean much, right? I think that's also another really fascinating aspect of this, the biology here.
Great, thanks so much.
Thank you.
Yukiko, I guess, and I doubt that you guys have done this yet, but like kind of more conceptually, I guess, feeding off of the, like what Alyssa was talking about with the stem cells and like the soma. I'm just wondering if this could be like a mechanism employed to like maintain pluripotency or something like that. So like,
I think so, because when you do the IPS, for example, right, and then clearly telomerase gets reactivated.
Right.
So I think similar things must be happening with this system of the RNA, copy number recovery. And with that said, I think I might want to actually bring up this, the interesting phenomena, or interesting, the fact that many people know, but have ignored. So, the laboratory mouse strain has outrageously long telomere, right? So that means, when, the people wanted to study the importance of telomerase, right? The typical laboratory mouse strain, if you knock out telomerase, they are fine for like 7 generations, right? So then, to really see the effect, that they had to actually, the breed the mice like 7 generations until you see the impact of the telomerase, But then, really curious thing is, still those, the mouse cells, the primary culture of the mouse cell that has long telomere, they undergo senescence. Okay, and then those cells, when those cells stop dividing, they still have huge telomere left. Why they are undergoing senescence, right? And then now I have to wonder, is that actually ribosomal DNA copy number, that hit the, the lowest minimal, right? So, because, if you need 2 things to stay, to keep proliferating, of course, if you lose one of those, that's more than enough for you to stop, right? So, I think that's another thing, that we have been speculating. Again, of course, we don't have any, the real answer to it.
Yeah, I wonder if you could, like, collaborate with somebody that works with, like, planaria or something like that, like a highly regenerative organism, and see if...
Yeah.
Like, maybe if you get rid of the... like whatever mechanism to maintain this rDNA copy number in planaria, then you cut them in half, and then it's like, no, they're just
That's really good point. I think we could do that. Yeah.
Does anybody else have any other questions?
Yeah.
Okay. Well, thank you for your time, Yukiko. We really enjoyed meeting.